esa diapositiva, eso. Y puntero, muchas gracias, Henry.
Estamos saliendo en vivo. Ah, perdón.
Sí, no, ya conversamos.
Ah, sí, entonces, ¿a poco le Yes.
una visita de una colega muy destacada desde Estados Unidos, desde la Universidad de California, en Santa Bárbara, que está trabajando en distintas cosas, ya lo vamos a presentar, pero el motivo de estar acá tiene que ver con actividades del magíster en paleontología, que se desarrolla desde el 2014 en la Universidad Austral, ya con varias camadas de estudiantes, y los proyectos que desarrollamos dentro de esta disciplina. Y quisiera invitar a mi colega y el director del proyecto de Fondesit, que hizo posible la presencia de la colega Vicky Goetze aquí en la Universidad Austral. Por favor, Cornelio, si puede presentar a nuestra.
Muchísimas gracias, Karen. Muy buenos días aquí aquellas y aquellos presentes, y también a nuestra invitada doctora Victoria Oelze. Voy a hacer la introducción en castellano, luego la charla será en inglés, y como sabemos también al mismo tiempo transmitida por el canal de YouTube de la Facultad de Ciencias. Algunas palabras sobre Vicky. Vicky misma también tiene unas diapositivas también hablando de su trayectoria, pero quiero destacar que Vicky Oelze es antropóloga biológica de la Universidad de Mainz en Alemania, y luego hizo un doctorado en el Instituto Max Planck de Antropología Evolutiva en Leipzig, también en Alemania, donde ya hizo o estudió la ecología isotópica, ¿ya? Primero en muestras arqueológicas, ¿cierto? Pero desde allá se ha cambiado a temas de ecología más actual, también en primates no humanos, entre otros chimpancés. Actualmente Vicky Oelze es profesora asociada de la Universidad de California de Santa Cruz en el departamento de antropología, ¿ya? Y ahí dicta clases en antropología biológica, sobre todo en el enfoque isotópico, ¿ya? Para mí es un gran placer también, junto con mi colega doctora Karen Moreno, ¿ya? Presentarles o tener esta oportunidad de tener a Vicky aquí presente, ¿no? Porque nosotros trabajamos en conjunto en el proyecto Fondesit sobre ojalá que salga así, hacer análisis isotópicos de unos fósiles de nutrimolados, ¿ya? Y para este fin está acá, primero para conversarles, mostrarles lo que se puede hacer en cuanto a los análisis isotópicos en la paleontología, arqueología, etc. Y luego también internamente conversamos también sobre cómo podemos adelantar con este proyecto nuestro, ¿ya? Sin mucho más agregar, aquí tenemos a la doctora Oelze, que nos va a presentar sobre insights into the past ecologies via isotope analysis.
Thank you. Okay, cool. Hi, everybody. Thank you so much for inviting me, Karen and Cornelius, to give a little talk here about what I know about isotope analysis, which I have been working with for close to 20 years now. And I've been working with a lot of different isotope systems. So what I thought that I could do here as a lecture is not so much like talking about my own work. I will also talk about my own work. But given we have so many students here in the audience, I also wanted to just give a general overview of all the things that we can do with isotopes to address past ecologies of various animals and also humans. So again, thank you very much for this invitation, and I hope the English that I'm speaking is going to be very accessible to all of you. If there are maybe some expressions where you guys know that people go like, what is she talking about? Please interrupt me, okay? Like, no problem, I am happy to explain. But I'm gonna not make this talk very technical, okay? I just want you guys to understand what an opportunity it can be to work with stable and also radiogenic isotopes. Okay, so just to get started, I wanted to say just a couple of words about myself, how I got into using isotopes. And so my path is a very roller coaster one, because as Karen just said, I started working on dead humans, then I moved over to working on African great apes, and now I'm working on making large strontium maps for entire continents. I work on archaeological horses, and now I'm also even invited to work on some Miocene fossils, which is, you know, relatively new to me. But the thing that brings everything together is the method that I'm using, right? So as a specialist in isotopes and isotope ecology, I can use the method that I'm good at and apply it to all of these different fields. And as you will see, like, even I've been doing like some plant physiology work, and it's like kind of all over the place. So I started doing my master's in physical or biological anthropology at the University of Mainz in Germany, because I really got obsessed working with human remains. And so had many opportunities to excavate mass graves and work with very, very early human remains from Germany, from the Neolithic, Bronze Age, Iron Age. And there already I figured I want to know more about the lives that past people and populations have lived. So isotopes was a very wonderful tool to actually look into the diets and also animal husbandry strategies that past populations used in Europe. So I started working on the earliest farmers that we have in Central Europe. Then at the Max Planck Institute, I continued this work for my PhD. I was in a very well-known archaeological science lab, where some of the best isotope people at the time were all working together, which was really great. But already during my PhD, and you can see that here, you see here there are a lot of skulls on the screen, but those are not human, but those are chimpanzee skulls that we had been receiving from the Congo. And so I actually started doing a lot of primatological work, studying first bonobos, but then also mountain gorillas, lowland gorillas, all kinds of chimpanzees, and working not only with skeletal material, but primarily also with hair, to look into the dietary ecology and niche partitioning of different great ape populations. So I continued doing that as a postdoc. So here's the team that I was working with at the Max Planck Institute as a postdoctoral researcher, where we studied the ecology of chimpanzees at 40 different field sites across Africa. And I was in this project responsible for isotopes. So mainly working with hair samples from 40 different populations to look at their dietary ecology, seasonality of diet, and also meat consumption in different chimpanzee populations. And then I seized the opportunity to go to the United States because there was a position opening in biological anthropology, tenure track position. And so I seized that opportunity to start my own lab, which is the PIMA lab, which stands for Primate Ecology and Molecular Anthropology. To reflect that I'm still doing both. I'm doing archaeological science, so biological anthropology, and at the same time, I also consider myself a primatologist. So I live in both worlds at the same time. Okay, so now what I wanted to cover today in my presentation here is, as I said, give you guys just an overview of all the wonderful isotopes that are out there. And I'm going to start with just a very basic introduction. What are isotopes? Because I don't know how familiar you guys are. Does anybody of you know what isotopes are? Good. No? Good. A little bit. Okay. We're just going to cover the basics here, so you go out of this lecture a little bit more informed. I'm going to start then with what we call traditional isotope systems. So those are really isotope systems that we have been using partially since the 1980s, 90s, very, very well established, particularly carbon and nitrogen. That's probably what you have seen in the literature before. Also oxygen. You as paleontologists, I probably know oxygen very well, so I will go over that relatively. Oh, that's a very, that's better, thank you. So we're going to go over those traditional isotopes, including strontium. So I work a lot with strontium isotope analysis, where we look at radiogenic isotope of strontium to reconstruct mobility and migration of past animal populations, individuals, but also humans. And then I'm going to switch over to the non-traditional isotopes. So actually these new isotope systems that are being developed over the last 10 years or so, and they have a lot of potential, particularly in the work that you guys want to do in paleoecology. And I'm going to demonstrate the utility of all of these different systems. You know, some examples from primatology, so working with living primates, paleoanthropology, so there will be some fossil hominins in there, and then also paleontology. What I wanted to say to start is the abstract said that I would also talk about sulfur and stable strontium. And like, there's just no way that I can squeeze this into 1.5 hours, okay? So this is already challenging to cover all of these. This is normally a lecture that I teach over more than, 35 hours over the semester. So I'm not going to talk about sulfur because I figured actually sulfur is not of relevance to you guys. Because we work with sulfur, we need organic material that contains a lot of sulfur. So that's bone collagen, hair. And I don't think that you guys can work with this in deep time. So let's just ignore sulfur. I actually don't like sulfur isotope at all. I know there will be some marine ecologists in the audience. So in marine ecology, sulfur is excellent, yeah, and very useful. But we're not gonna cover it today. And then I'm also not gonna go about stable strontium. So that would be the ratio of strontium 88 over 86. But it's very similar actually to zinc and calcium, so bear with me there. Okay, so let's just get started with the absolute fundamentals. What the heck are isotopes actually? Okay, so per definition from good old textbook chemistry, isotopes are the different forms of the same chemical element that actually are the same in the number of protons that they have in their structure. So they have the same atomic number, and you find them in the same position in the periodic table, but they have different numbers of neutrons in the nuclei, okay? And that is just the beauty of nature. Nature is kind of a little bit chaotic, and so you find different isotopes of all elements pretty much have different isotopes. So you can see here at the example of carbon. So we have carbon-12, carbon-13, carbon-14, and carbon-12 and 13 are stable. And you can see that carbon-13 simply just has one more neutron in the core. So these are the different isotopes of carbon, but actually this one you might have heard about, carbon-14, is not a stable isotope. This one is a radioactive isotope, right? So it undergoes radioactive decay. But the other two isotopes, carbon-12 and carbon-13, they are stable, okay? So they're naturally abundant in the environment. They have nothing to do with radioactivity or anything like that. As I said, most elements have some version that has one more neutron. And so you can see also that the isotopes are distributed in nature very biased. So carbon-12 is almost 99% of all carbon is carbon-12. That's like the standard carbon. And only some have this extra mass, which turns them into carbon 13. Okay, so carbon 13 is very rare. Just like, for example, in nitrogen, nitrogen 15 is very rare. Yeah, only 0.3% of all the nitrogen is actually nitrogen 15. Everything else is just a standard nitrogen 14. And so when we look at isotope ratios, we actually look at this association between how much of this unusual heavy isotope is there in relation to the normal light isotope of a given element. Yeah? And we express this here with the delta notation, so the Greek delta, and in per mil, because like the reference standards that we use during analysis actually produce very small numbers, like 0.00, and that's why we just multiply it by 1,000. And that's why you see a lot of the data that I'm going to show you today is like always in per mil, yeah, not percent, but per mil, because otherwise the numbers will always be too tiny, and that would be very annoying. Okay. So this is the standard isotopes here. It's the same for sulfur, where we look at, actually sorry, there's a spelling mistake here, it should be the other way around. I always forget to fix that error here, so it's sulfur 34 in comparison to the more abundant 32. Oxygen is the same. Strontium we refer to a little bit different, and I'm going to get to that later, because in strontium, one of the isotopes, strontium 87, is actually not considered just a stable isotope, but it is radiogenic. So it is a stable isotope that is born out of radioactive decay of a different element. So to just acknowledge that fact, we don't use the delta notation for strontium. But okay, let's get into the actual interesting stuff, how we measure these isotope ratios. So how much of this unusual, more heavy isotope, for example, carbon 13 or nitrogen 15, do I have in a sample material? For example, tooth enamel, or maybe plant material, or insect chitin, whatever you want to look at. So the beautiful thing about isotope analysis is most of the time we only need very small amounts of material. Yeah, we're talking about micrograms or a couple of hundred nanograms of material. So that's nice, keep that in mind, yeah? So we can almost consider in a lot of cases isotope analysis as minimal and destructive, yeah? So what we do is we work with isotope ratio mass spectrometry. Has been around since the 1920s, okay? So like these instruments are really old in their technology. So it starts that we have a sample combustion interface where the sample material is turned from solid form into gas, most of the time by combusting it, by burning it. And the sample material that is then in gas form will be electrically charged and then sent through an electric field that basically just accelerates the elements or the atoms to basically catapult them against a pretty massive magnet, yeah? And so these magnets are actually pretty big, like you can see them in the instrument, they're often like a really big chunk that is then the magnet. And the magnet, the only job of the magnet is to deflect the different isotopes of the elements by their mass, okay? So anything that's larger in mass will be a little bit slower to react to the power and the force of the magnet. And so you will have like this deflection here that separates them by mass. And on the other side, we have detector cups that then will recognize the charge that is arriving in the different detector cups and will then tell me how much of the different isotopes I had in the sample that I shot through this mass spectrometer. And so actually the technology is relatively simple, and a lot of isotope ratio mass spectrometers are not that crazy big instruments. They can be relatively small. So here's like some very standard setup that we can use for carbon and nitrogen, sulfur, oxygen. But then some of these larger elements, like heavier elements like strontium or also zinc, like the metals, we actually need a little bit of a bigger machine. So here we use inductively coupled plasma mass spectrometry. And they are really big instruments. Sometimes, like for example here, the Neptune that I work with for my PhD, they're like as big as a truck, right? So like you need also the infrastructure to have an instrument like that. I personally don't like this particular instrument because they're pretty faulty. They often like break and don't work. And very difficult to find the error. And during my PhD, our ICP-MS was actually broken for 13 months when I just started my PhD. But it was a great opportunity because I had 13 months where I was waiting, and that gave me the opportunity to move into primatology and start working with primatologists on completely different things than archaeology. So it can be an opportunity to, have to think, what else can I do as a scientist? Okay, so the basic underlying principle for the first isotopes I want to talk about, namely nitrogen and also carbon and also oxygen, they are pretty much stand for this idea that you are what you eat, or you also are what you drink, right? Like things that are actually going into your body will change actually the isotope ratios of your body tissue. And so the different elements that we're going to cover today, a lot of them actually go into the body through the diet, okay? So that's you are what you eat. So particularly carbon and nitrogen make their way through the diet into our body. And of course, oxygen has other pathways, for example, drinking water and also the air that we breathe. But the air that we breathe, the isotope ratios of air have been stable pretty much. So that is not really of interest, but the water that we drink is really indicative of the isotope ratios that we actually have in our bodies, in our skeletal tissues. They directly relate to the isotope ratios of the water that we were drinking throughout life. And so these elements go into our body. Well, guess what? They also have to leave our body at some point, right? And so they're excreted in different ways, particularly through feces and urine. That's mainly the pathway that a lot of these isotopes, the elements and also the isotope ratios that they have actually pass through. But something actually happens in the body for a lot of these elements, which is what we call isotopic fractionation, okay? So many, but not all of them, okay? So actually strontium does not fractionate at all in a noteworthy way. These isotopes actually fractionate in our body because they have different mass, okay? So if an element is a little bit heavier, an isotope, sorry, is a little bit heavier, for example, carbon-13 is heavier, nitrogen-15 is heavier, sulfur-34 is heavier. That difference in atomic mass can actually influence how they pass through our body, because you can picture it in a way that like the heavy isotope preferentially gets incorporated into body tissues. So it's kind of like almost like in the mass spectrometer, it's a little bit lazier to move through, membranes or be mobilized in the body. And so the lighter isotope is often the one that gets excreted preferentially, okay? So your body is always isotopically heavier than the food that you were eating. And given that we've been measuring this kind of stuff since the 1980s, we really know how much this isotopic offset or fractionation is. And this is particularly important for nitrogen, but also for carbon, oxygen, and as you will see later, also for calcium and zinc. But they're a little bit freaky. They actually do the reverse. But I'll get to that. But isotopic fractionation is the underlying mechanism for a lot of isotope systems, except for strontium. Okay, so let's start with nitrogen and the stable isotope ratios of this element. So nitrogen is very, very abundant in any kind of organic material. mainly associated with proteins, right? So if you're thinking about anything that contains a lot of protein, it will have a ton of nitrogen in it. And in nitrogen, this isotopic fractionation, this isotopic offset between what you eat and what actually is incorporated into your body tissues, including your skeleton, even your teeth, is about 3 per mil, and relatively reliably around 3 per mil. So the actual number is more like between 2 and 5 per mil. There's some variation there, but there's always like an offset that if you are an herbivore who's eating plants, predates on plants, you will have about 3 per mil higher nitrogen isotope ratios than the plants you are eating. And then if you're a carnivore who is eating those herbivores, that carnivore will be about 3 or like 2 to 5 per mil higher than those herbivores. So it's a really cool system. And as you can see here, carbon also has a little bit of an isotopic offset, but it's relatively small. It's just 1 per mil. But you will see a lot of plots in the literature where people look at the ratio or the association of nitrogen isotopes and carbon. I'm going to get to carbon in a little bit because carbon is actually way cooler than just this. But if you think about nitrogen isotopes, you really have to think about, because of this trophic level effect, so with each step in the food web, the isotope values go up. It can say something about meat consumption, differentiate an herbivore from a carnivore, which can be cool if you really don't know what a certain animal was eating, as you will see later, what a fossil hominin was eating. But it also indicates breastfeeding, right? Because if you are being nursed by your mother, you are, in fact, just a little carnivore eating your mother's body tissue. So you're isotopically enriched by about 3 per mil compared to your mother. And we can see that in the archaeological records. It's very, very important to assess at what age children are weaned from breast milk. And that's something that I actually study in all grade 8 populations now. Looking at like when chimpanzees, bonobos, and mountain gorillas are weaned from breast milk, which says a lot about like interbirth interval and all these kind of reproductive success of females, blah, blah, blah. Okay, so that's why we see here a little icon indicating milk and meat, because both are just animal proteins and animal tissues. So the reason why I'm telling you this, because normally in a talk in front of a paleontological audience, we would not talk about nitrogen, because nitrogen is not really abundant in the skeletal materials that you guys work with, right? But there was just like recently a huge innovation by my friend Tina Lüdecke and her team, including her postdoc Jen Leitlichter. They are actually now able to measure the tiny amounts of nitrogen that is in tooth enamel. It's like just a couple of percent of hydroxyapatite of tooth enamel. There's some little amounts of protein actually in there, and that nitrogen can actually be captured with a new method that was developed at Princeton University a couple of years ago. It's called the oxidation denitrification method, where you literally hand over about 5 milligrams of tooth enamel to some very specialized bacteria that are very well kept, like almost like the most precious animal in a zoo, and well maintained. These bacteria actually then eat away the tooth enamel, and they are able to actually isolate the nitrogen, and they fart and burp out the nitrogen, and then you kill the bacteria, and you take up the gas that these bacteria are producing, and that's enough to conduct isotope analysis on a mass spectrometer. So it's really crazy cool stuff. And that actually allows us now to measure nitrogen isotope ratios in fossil material. So the first applications, I don't know if you've heard about it, were in like some gigantic fossil shark species, where they showed like, yes, these gigantic sharks were eating other animals. Okay, that just confirmed like what we already knew. But it's still like really, really cool that we can like now look into past food webs and answer some questions about trophic level of certain past animal species. Okay. In the case of the work that Tina Lüdecke and her team at the Max Planck Institute for Chemistry in Germany have been doing, they looked at several extant, so like living animal communities in Africa, but then also immediately applied it to the fossil record of faunal communities in Africa. And you can see here the nitrogen isotope values. So here you can see on the y-axis are the nitrogen isotope ratios. So the resolution is really not good here. I hope you can see the graphs okay. But you can see that the herbivores here, they always have lower nitrogen isotope values than the carnivores. So in this case, like some larger felids where we know, of course, they were like eating meat. And this offset is absolutely beautiful in different ecosystems. It's a little bit different, right? So here in this case, it's around 3 per mil, in the upper case, like 3.7 per mil. It's like somewhere around 3 per mil, that difference between herbivores and carnivores. I should indicate here too that you might look at these elephants and go like, oh, that is very, very weird, huh? And that is actually one of the biggest mysteries that we have also in archaeological science more widely when we study Neanderthals, where we know that Neanderthals were highly specialized in eating or in hunting woolly mammoth and like this really like incredible megafauna. And woolly mammoths, their nitrogen isotope values are always very, very high. And it's not because they were eating meat, right? Like we don't really think about like elephants to eat meat, but they're eating something weird that has high nitrogen isotope values. Or maybe it's like their gut microbiota that causes more fractionation between the diet and their body tissue. We don't really know, but it's something that you will see in the literature over and over again, that the elephants always look weird. And in the case of Neanderthals, it actually causes them to look even more than an apex predator. So Neanderthals are always higher in nitrogen isotope ratios than the cave lions and the wolves and other large carnivores in their animal community. And it's just because they're the ones that are eating most mammoths, which is really, really cool. Okay, so Tina's work is absolutely incredible, because the biggest question that we paleoanthropologists or anthropologists have is how meat eating actually evolved in our lineage, right? We think that brain size increased because at some point we were eating higher quality foods. And we know probably later it has something to do with us starting to cook, getting more nutrients and more energy out of lesser quality food. But in the beginning, it's probably really meat eating, right? And so in a lot of fossil hominins, we really wanted to know what trophic level do these hominins have. And the isotopes that we could measure so far were carbon and oxygen, and they don't have an answer to this question because they don't relate so much to meat eating. So now Tina is actually throwing this new method, nitrogen isotope analysis, at all kinds of hominins she can get her hands on. And this was just published a couple of months ago in Science. And so this is the first study showing nitrogen isotope data for fossil hominins, in this case Australopithecus from the cradle of mankind in South Africa, so the site of Sterkfontein. And you can see here in pink is the data of these Australopithecines. We didn't go into like the actual species because that's apparently debated, and we don't need to get into the debate, right? It's like some fossil hominins, and they're together with some other primates and herbivores, and then you can see up here are the different carnivores from the same site. And you can see that those hominins shown in this pink color, well, they don't really overlap with the carnivores, do they? Except for maybe like 1 or 2 individuals here that have higher nitrogen isotope values around like 9. That might overlap here with some of the larger felids from the same site. So basically the conclusion is that at least for these guys here from Sterkfontein, we don't really have evidence that they're eating a lot of meat, except for maybe some of these higher nitrogen isotope values in 1 or 2 individuals. So that was also like the conclusion of the publications, like we don't really see them being like meat eaters in their species community. They are probably largely plant eaters, maybe like eating ripe fruit and stuff like that we see in a lot of great apes today. Okay, I just wanted to get over to the topic of breastfeeding, right? Because like you were very excited about hearing that little babies are basically carnivores in a way, liquid carnivores, right? So we actually did a study in collaboration with Tina at a field site that I've been working at now for many, many years. I mainly work on the camera trap material there, so we're looking at like the behavior of the chimpanzees and their termite fishing behavior. So like again, I do a lot of different things. But here I sampled all the yellow baboons that we have a skeletal material of that was just like found over the years since these chimpanzees have been studied and the baboons have been studied. And then we also had some skeletons of free-ranging chimpanzees from the same site. And here I sampled tooth enamel, like really small amounts of tooth enamel, for nitrogen isotope analysis of enamel. It's a super new technology, I said, just around for a couple of years. And this is actually the first time that we compared early forming teeth, so early forming, in this case, our first molars, to the isotope values that we measured in third molars that form certainly after individuals are being weaned from breast milk. And you can see here that we have very, very clear isotopic offsets for all of these different individuals of both baboons and also chimpanzees that show this breastfeeding signal are these gray squares that are like the early forming teeth when those monkeys and apes are still breastfeeding. And so this is just to say that with this study, we still need to write up the manuscript and like eventually publish it. So this is something I worked on with Sven Brümme, who's a PhD student of Tina. There's really huge utility of this method to look into breastfeeding behavior of animals that have been, that are dead, but obviously we can look into the fossil record too. And we can go pretty deep into time, because if you have tooth enamel, you will have trace amounts of nitrogen in there, and you can look at this in the fossil record. Okay, so let's move away from nitrogen, and again, if you guys have any questions about nitrogen isotope analysis or are interested in applying that to maybe the fauna that you are working with, let's talk about that. But let's move over to carbon. I already said that there's like a little bit of a trophic level effect, so like 1 per mil, but it's really not worth talking about, I think, because actually in nature, plants are the main driver of variation in carbon isotope ratios in animals that are living in different places. And so the largest difference that we have is actually between C3 and C4 plants. And isotopes of carbon can also help us to differentiate if an environment was a forested environment with a lot of like plant biomass, or if we have like an open grassland or with relatively little canopy cover or vegetation density. So we can differentiate between these very different biomes very, very easily, actually, with carbon. In humans, of course, that is more related also to our obsession with domesticating C4 plants, right? So humans love cereals, obviously, and some of the cereals that we have domesticated over time are C3. Actually, a lot of them, like maize and sorghum, are C4 plants. So that is something that I personally am very interested in my archaeological work. Okay, so the main underlying fact here that we have in carbon isotopes influencing the carbon isotope values being influenced by the physiology of plants is related to photosynthesis. So C3 plants are the most abundant photosynthetic pathway on our planet. So about like 97 to 98% of all the plants out there are C3 plants. That's like the baseline, yeah. And they use the Calvin cycle to form sugars during photosynthesis. And C4 plants are actually the ones that are very unusual. And they produce these really, really high carbon isotope values by comparison to C3 plants because they use the Hedge-Slack photosynthetic pathway that discriminates less against carbon-12. So the isotope values are always higher. And these plants have adapted to more dry environments that are very hot. And so basically C4 pathway is just like a photosynthetic adaptation to living with water shortages because it avoids the evaporation of water from the leaves. And so these plants are always associated with certain environments. But here I kind of like wanted to stress, in case that we have some of you maybe working on some fauna that is not just like falling into this dichotomy of, you know, forest or open savanna or prairie, as it's sometimes called in the Americas, or I guess like pampas, and also to here. Because there's actually already a lot of variation going on just within the C3 world, right? Like the vast majority of all plants being C3. And I would like to argue that actually the variation in carbon that we see within C3 is just the same as between average C3 plants and C4 plants. And so this is actually something that I have looked into more detailed in my own work, because this is related to the so-called canopy effect that was first discovered in the 1990s. So this is not new, okay? So since the 1990s, we know that plants that are in a forest, trees that are very high, or plant material that is very high up in the forest canopy will have high carbon isotope values, and the plants that are growing in the understory, like on the forest floor, that they have very, very depleted, so very, very negative carbon isotope values. So in my own work, I wanted to look into this a little bit more detail, but the general effect, again, has been very well understood, okay? So again, we have in the understory here low carbon isotope values that are probably related to that there's very little air circulation in the lower parts of the canopy. If any of you have ever worked in a tropical rainforest, I just come from being in Costa Rica for a couple of days in a jungle there, and at the canopy, lower canopy, you have very, very little light that comes through, and also very little fresh air that actually cycles through that part of the forest. And this recycling of carbon by all of the organisms that are living in the understory, including mainly microbes, causes a lot of isotopic fractionation and lower and lower and lower carbon isotope values. And so generally, for your work, don't always think, oh, it's like C3 or C4. You will actually also see that even in the absence of C4 plants, you can see this isotopic difference between a very open habitat environment and a more closed environment with a lot of canopy cover. So I wanted to get into my own work that relates to the canopy effect, because this is actually the first project that I did when I went to the US and suddenly had no boss anymore. So that's kind of like maybe interesting for the younger ones of you. It's really nice to not have a boss anymore, and you can actually let your scientific creativity flow. Well, ideally, you can already do that under the supervision of your advisor, but it's really nice to actually be able to just say like, hey, I want to answer this question that I had in my mind for such a long time. So I had been working on chimpanzees in Tai National Park in West Africa already for a few years. And I was always very struck by the intensity of the canopy effect in this particular environment. It's a primary tropical lowland rainforest. And literally when you're on the lower levels of the canopy, it's just very, very humid and there's no fresh air. And you can literally feel that fresh air doesn't really come into that part of the environment. Whereas you can always see the upper part of the canopy being affected by winds and stuff like that, so there's a lot of fresh air blowing through there. So for this project, I worked with an arborist. So already from my previous project, I learned how to climb trees using rope-based tree axes. because I needed to climb into chimpanzee nests to collect hair from chimpanzees. And tree climbing is really, really fun. So I was like, okay, my first big project will be about tree climbing. In this case, looking into the plant physiology and the vertical stratigraphy of isotope ratios in a primary tropical rainforest. And so we sampled trees of different height. So you can see here we have a couple of trees that have they're very, very tall. They're these beautiful emergent rainforest giants, up to 40 meters tall. And we sampled leaves of these trees at 1 meter intervals. So we climbed all the way to the tip of the tree, and then every 3 meters we took, every 1 meter we took a leaf sample, and also for each leaf sample measured how much light was actually reaching that particular leaf. And you can see we tried to really go through the entire canopy and also sample some terrestrial herbaceous vegetation. And all of the plants that we sampled here are actually relevant in the diet of the chimpanzees at Tai, but also different monkeys that are being studied there for a long time. And so then we did carbon, oxygen, and nitrogen isotope analysis to see, like, well, how strongly expressed is this canopy effect actually? And so here are some of the results. So again, like these lighter colors are these big emergent trees, and you can see height is still here on the y-axis, so the more we're up there, the higher in the canopy are the data points that we collected. And this PAR here is actually photosynthetic active radiation, which is a fancy way of saying light, yeah, so it's light availability. And actually we had a very, very strong association between height and how much light is actually penetrating leaves, you know, obviously, but it was nice to show that. And the canopy effect here, so that with the higher you are up in the canopy, the higher the carbon isotope values are, and vice versa, was super, super strong. And that was great, because that's what we wanted to show. And we also wanted to show that it's not just like up versus down, but it's really like layer by layer by layer, these values are becoming depleted the lower you go. But the cool thing was that we could also show it for oxygen, which had been shown before, but again, it just this dichotomy between high and low. We really showed that it's a really lovely gradient. And for the first time, we actually also showed it that it's even doing something with nitrogen, which we don't understand, okay? I'm an anthropologist. I never had plant physiology classes. So that is something that like some plant folks need to figure out, like why we actually see this gradient even in nitrogen. Because to me, it actually makes no sense, but I think some people are already working on it. So the canopy effect is real, okay? And it's really, really pronounced both in carbon and also in oxygen. And so that is actually really cool, because it can tell something about in what height in the canopy certain animals are having their home and are utilizing their food resources. So these are toothy enamel, oxygen and carbon isotope data, and then also nitrogen, back then only measured in bone collagen, from all these monkeys that actually live in Tai. And we have some monkeys there that are mainly in the understory, like mangabees, for example, and then we have the colobines that are like up in the tippy top of the canopy, and then we have several monkey species that are kind of like in the intermediate levels of the forest. And so here we could really show that carbon and nitrogen are both really affected by what these animals are eating, you know, like the trophic level. Are they eating insects, or are they eating tree sap, or are they eating leaves or fruit? But oxygen, as you can see here, really these pink dots here are the monkeys. So oxygen really tracks beautifully in what vertical position in the canopy these animals are feeding. So maybe keep that in mind too, right? Oxygen can say a lot about the canopy effect and about where animals are actually hanging out in a certain forest environment. And so let's talk a little bit more about oxygen. So oxygen isotope ratios are often used in combination with carbon isotope ratios in paleontology and also paleoanthropology, because until recently they were the only ones we could measure in tooth enamel, because there's a lot of carbon in tooth enamel and there's a lot of oxygen in toothed enamel. And so this here is a very nice example from a very, very early African hominin, Ardipithecus ramidus from Ethiopia, about 4.4 million years old, probably one of the earlier ones in our larger family tree, being a bipedal ape. So something that has descended from the trees and probably also is still going up in the trees to feed at night, to rest at night for safety, but otherwise like also terrestrial, kind of like is reconstructed to look as handsome as that. And so Tim White from UC Berkeley and colleagues, they did some, as you see here, carbon and oxygen isotope analysis on Ardipithecus that you can see here as this yellow star here, in association with the other animals that were found at the Aramis site where Ardi was discovered. And you can see often the most helpful way of looking at data like that, if we're going so far back into time, is often looking at the whole animal community. Right? Because like some of these animals, they still have like living counterparts today, where we know quite a bit about their ecology today. And so you can really see here how Ardipithecus is clustering with a lot of other forest-dwelling animals, including like some extinct primate species. And is very, very different from a lot of the herbivores that were found at the site that seem to be C4 plant savanna grazers. Yeah? But what I want to say here is that the oxygen really brings into this extra level also saying something about where Ardipithecus, or that Ardipithecus is falling within this rainforest environment, in the presence also of these open landscapes where these larger herbivores are grazing. And so that is actually really, really cool. And I did like quite some work now on the teeth also of living chimpanzees. And actually Ardipithecus is very, very similar in both carbon and oxygen to chimpanzees in these mosaic landscapes today, where we have like open environments adjacent to gallery forests along rivers and stuff like that. So it's kind of cool that we have like a 4.4 million year old hominin that we can reconstruct what they were up to and what biome they were living, just using 2 isotopes, and that's it. Okay, so oxygen, let's talk a little bit more about oxygen. So I hope you will remember until eternity that oxygen has a canopy effect. That is like something that a lot of people don't really talk about. It's very different in open environments compared to forested environments. But normally when we talk about oxygen isotope values, the underlying thing that we're looking at is twofold. It's always about evaporation of water and condensation of water. So evaporation of water is always, if you think about large bodies of water, like let's think oceans, yeah? Particularly when we think about oxygen on a global scale. And then condensation, always we can think about precipitation, rainfall, yeah? And during evaporation and condensation, oxygen fractionates, okay? So when it evaporates, oxygen 16 is lighter than oxygen 18. And so oxygen 16 is the first one to say like, oh yeah, I'll turn into gas and I'll rise up to the sky and form clouds. Whereas oxygen 18 is more like, oh, I'll stick around and I'll stay here for a little bit more longer and wait for higher temperatures to kick in, that I also get mobilized. And the reverse effect is then happening when we have condensation. So when clouds actually turn into, when clouds precipitate and rain, their oxygen is heavier and is being more attracted by gravity and says like, okay, I'm going to go first. I'm going to rain out of the cloud first. And so we can see really interesting patterns emerging on regional scales that always have something to do with rainfall, where the water also comes from that we consider precipitation? So are we in a coastal environment where the precipitation coming from the oceans is relatively new, right? Has newly evaporated from the oceans, or if clouds have been traveling for a very, very long time, and a lot of the oxygen 18 has already rained out hundreds of kilometers before. So we have this coastal versus inland effect when we think about environments, and it generally, oxygen is always saying something about how hot and dry or wet and cold is it. On a local scale, but particularly on a global scale. And so the global scale, again, I think is really boring for you guys, because you will be very, very well aware of that, right? That oxygen isotopes is the key tool that we use to reconstruct the global temperature over the past. So we use ice cores to get really, really fine resolution climate data about the global climate for the last 100,000, 200, 400,000 years or so. And then if we want to go deeper into time, we actually work with marine sediment cores and the foraminifera in there to go back millions of years to reconstruct past climates. And so when we think about the global scale, again, you probably already know this, but maybe just to remember you that these oxygen isotope stages that we work with are always related to the fact that a lot of the Earth's water in glacial periods, so when it's really cold, a lot of the oxygen 16 is just trapped in ice and cannot re-enter into the ocean. So that's basically the main thing that we're looking at. Some of the oxygen, maybe oxygen 16, just being, packed away until we have like a warm period kicking in again, and all of that oxygen 16 being released into the oceans again. Okay, that's all I have to say about oxygen. So oxygen really relating to the drinking water we eat and saying something about the climate at a given place where we are. Okay, let's move away from the typical stable isotopes and let's talk a little bit about strontium. So strontium isotope ratios are actually like my favorite isotope because they're so incredibly reliable. So when I told you about nitrogen, I said like, oh yeah, nitrogen always fractionates by like 3 per mil, and so we can estimate like how much meat was eaten or something like that. Well, but then in fact, it's actually like more somewhere between 2 and 5 per mil, so we don't really know, there's some variation, right? So strontium is so honest in comparison because strontium actually does not fractionate between what you eat and your own body tissues. But everything in a food web in a certain place will have the same strontium isotope value. And that strontium isotope value is only linked to the strontium that is bioavailable in our environment, and it's always associated with the underlying geology or bedrock. So in that way, it's like very, very honest, and it gives you an isotopic fingerprint of place, of a location, of a geology, okay? And so strontium-87, as I mentioned before, is a radiogenic isotope, and that term can sometimes be a little bit confusing because like a lot of folks are like, well, does that mean it's radioactive? No, it's not radioactive. Strontium-87 is born out of the radioactive decay of rubidium-87. And so you have more strontium-87 in a place where more time could elapse, so you had more radioactive decay of rubidium, and that is why we have in older bedrocks, we have more strontium-87, because more time could pass and for radioactive decay of rubidium. And so if we are in a place where we have very old bedrock, we have very high strontium isotope values because we have a lot of strontium-87 where bedrock is very, very old, geologically old. And we have very, very low strontium isotope values, so very little strontium-87 in places where the bedrock is super new, for example, in volcanic areas, right? So if you look at Hawaii or something, where you literally have bedrock forming as we speak, right? Because we have like one of the volcanoes being, erupting right now, you will have strontium isotope values that are ridiculously low, right? And so how it works is that the strontium is a trace element that is not essential. So our body doesn't really need it, although some people will say it's like helpful for osteoblasts to actually produce bone material or hydroxyapatite, but it's not considered an essential element. But it is chemically very similar to calcium. So we also call it's bioequivalent to calcium. And so if you are living in a given place, the strontium will weather out of the bedrock, right? Well, like a lot of other minerals will become available for plants to take them up. Plants will actually take it up accidentally instead of calcium because it looks chemically so similar. And so trace amounts of strontium are already assembled or taken up by plants from a local soil environment. And from there, it just passes into the food chain, but again, without isotopic fractionation, so without anything really happening to it. So the isotope values of strontium in a given place will be the same in the plants as in the fauna that is eating those plants. And this allows us to use strontium isotope values in body tissues, such as teeth and bones, to say something about where an animal was living. So where the plants or the other animal foods were located that they were eating. So it says something about, or gives an animal an isotopic fingerprint of the geology. And that allows us actually to understand if an animal is changing location, changing its place in terms of geology, and that allows us to reconstruct mobility and migration in the archaeological record, but also in modern animal communities. So people, for example, are studying reindeer migration using strontium, and I work a lot with strontium in the archaeological record as well. So here I wanted to give you a paleontological or a paleoanthropological example of how strontium can be used. So this is a really cool study that was published by Sandy Copeland, who was a postdoc at the Max Planck when I was doing my PhD. So Sandy actually trained me in all the lab work associated with extracting these tiny amounts of strontium from tooth enamel, which is really fun, wet chemistry approach. And she applied it to hominins from Sterkfontein. So we're back at Sterkfontein in South Africa, in the cradle of mankind, and also swartkrans. So here they were looking at Paranthropus robustus and Australopithecus africanus from these very, very early sites in South Africa. And so here, the interesting thing is they are looking at strontium isotope values. So we have here a range of strontium isotope values. Some are higher, some are lower. So higher values will always indicate older bedrock that an animal was living on, and lower values a younger bedrock. But the variation in isotope values in these hominins itself already says that they were growing up in different places, right? Because otherwise they would all look the same isotopically. And so the interesting thing that they did here in this paper was it is impossible to sex these fossil specimens, particularly if we just have a couple of teeth. But they were here saying that smaller teeth, so that are generally just small in size, that they might be coming from females, and that larger teeth are more likely to come from males. If we apply the same sexual dimorphism that we see in other living apes and also in humans. And so here they showed that you can actually see the ones that are potentially female, they're shown in white, those are the smaller teeth, that they seem to be coming from not the geological area that is identified here based on local strontium isotope values that they measured in local plants and other things in the Swartkrans and Sterkfontein area. Okay, so this blue zone here, these values in this range would be local, and this gray zone here would be non-local. And that's kind of cool because, like, you know, then they made a big thing out of it and it was published in Nature saying, like, you know, like, oh, maybe these fossil hominins are like male philopatrics, so you have like the females being the sex that is more mobile and joining other social groups for reproduction. What I wanted to emphasize here is for any kind of work in strontium, if you guys are interested in doing that, I think, again, I love strontium. It's like so widely applicable to so many questions, and it's so reliable and honest. But what you need to do is you need to sample the crap out of the study area that you're working in. Yeah. So here we have Swartkrans and Sturpfontein, and you can see Sandy was using a geological map of the larger area, and all these red little dots, I hope you can see them, are locations where she sampled plants and also snail shells and stuff like that to establish what values in strontium would we find in this larger area, right? So it's often about finding out, like, how does local look like, to then say, like, oh, this animal has a strontium isotope value that is non-local. Yeah? And so this is something that I have been working on already in my PhD. I mapped like larger areas of southwest Germany because I wanted to know if some of my archaeological humans from the Iron Age and the Bronze Age, if they're considered local or non-local. But the biggest project that I've done in my career so far is actually this one, where we built a strontium isotope map of sub-Saharan Africa. This was a project I worked on for 12 years. So that's like maybe something also to consider for our younger colleagues here. Sometimes you just like have to be in there for the long run. So we started sampling in 2012, just getting from any area that I could get my hands on, would get like plant material, ideally targeting all these different geological areas on the African continent. And then in the later, last like 5 years or so, I also just reached out to hundreds of archaeologists that were working across Africa, but also plant biologists, to just say like, hey, do you have samples? I just need a couple of grams. I need 2 grams of plant that you don't need anymore. As long as it has a GPS coordinate and it was not in an urban environment where you have contamination issues, can you please send it to me? And that's why this paper that we published in December of last year, with this isoscape, so it's an isotope landscape of sub-Saharan Africa, has about like 116 co-authors because like so many people contributed samples to this project. But here, so we used, it's actually like only around like 2,000 data points for sub-Saharan Africa. It would have been great to do more, but you have to maybe keep in mind that each analysis of strontium costs around like $100. So it's like something that is, you know, you need the financial means to actually do the work, and it's kind of tedious lab work too. But we have now, since a couple of years, like Clement Bataille and his colleagues, they developed a new modeling approach called random forest, where you can actually not just like say like, okay, the isotope values look like this, and then do some very, very basic extrapolation of your data. These random forest models integrate all kinds of maps related to geology, sand deposition, soil pH, you know, ion mobility, and all these kind of things. Anything that can influence strontium isotope variation in a given place is integrated in this model. And you can see that we have incredible variation in the African continent. So these red areas here, red or orange areas, are areas where the strontium isotope values are really, really high, incredibly high. I actually had never seen strontium isotope values beyond 0.7. So you can see we have areas where it's 0.8. And that's just because the geology in Africa is just so incredibly ancient. The ancient bedrock being really pushed all the way up. What we also did here, which is kind of like new, and I'm super happy to talk about that with anyone who's interested, is you see here there are areas that are white. Some of them are just because it's like large lakes, but the other areas that are white is like where our model was unable to predict into that area, right? And normally these isoscapes will predict into those areas where they don't have data anyway, and we just decided that we will not do that. You can see a lot of sub-Saharan, of the Sahel zone up there is white, and it's just because those areas are actually inaccessible in terms of safety concerns for doing field work there. So we hope that we can fill this gap in the future. So maybe just like the last thing I wanted to say here is very, very away from the research that you guys are doing, but I mainly am using this map, and that's why I also built this map, is to look into the origins of people that were subjected to the transatlantic slave trade. So human remains that are found in the African diaspora in the Americas, in the Caribbean. I work on a site in Portugal, and using this map, I can actually say a lot about where people originally came from, with a huge potential for the ability in the future to sending these African ancestors back home. So in terms of repatriation, this data is very, very important. And genetics can go only so far, and combining ancient DNA with strontium is really, really cool in terms of thinking about repatriation of people from the African diaspora. Okay, and for you guys, much more interesting is that we can actually apply strontium isotope analysis also really, really deep into time, because we can measure strontium in tooth enamel. Remember, strontium is bioequivalent to calcium. You have tons of calcium in tooth enamel, so you have tons of strontium in tooth enamel. So if you have like dinosaurs or other like big critters like that have good tooth preservation, well, you can pretty reliably measure the strontium isotope ratios of that animal in their teeth. It's really not rocket science to do that. I just picked up here one example. Often we can combine strontium with carbon and oxygen. So this is a really cool study here with some dinosaurs from North America, I think an area between Alberta and Montana, where they then used carbon and oxygen to show this migration between coastal and upland regions of these hadrosaurs, and also strontium to actually just show, if you look at, am I pronouncing that right? Hadrosaurs, would you say that? Hadrosaurs. They're shown here in blue, so you can see there's a lot of variation in strontium in that species as compared to the other ones. And so the authors just concluded, even without having a strontium isotope map of the area, to say like, these guys are very mobile, they're going places, whereas the other taxa are more localized. They're born, raised, and dying in a very, very similar geological area. And that's kind of cool, right? Like, that's kind of cool to say something about like the life ways of animals that are gone for a goddamn long time. Okay, so I still have a couple of moments to get into zinc. Okay, so zinc isotope ratios, and followed by calcium, and then you guys are done, okay? Like, I know it might be a little bit exhausting to listen to all these different things. So zinc isotope analysis is relatively new, was pioneered by one of Cornelius and my colleagues from the Max Planck Institute for Evolutionary Anthropology, but she actually did her PhD at the University of Lyon with her supervisor Vincent Balter, who I've been collaborating with now over the last few years as well, because like, if you want to do some of these cool non-traditional isotopes, there are very few labs that actually can do it. So keep that in mind, and also if you are interested in getting connected to those labs, please come talk to me, because they're always excited about all kinds of new projects. So the interesting thing here is, so Claudia Joen had to actually do, work with samples from like feeding experiments, but also from modern humans where you have like known diets, to establish that zinc isotope ratios fractionate in a very, very interesting way. So the difference between what you eat and how it looks like in your body tissue. Remember, in nitrogen, was like the nitrogen isotope values would go up by 3 per mil, or 2 to 5 per mil, 3. In zinc, it's super interesting, it's the reverse. So the higher you go up in the food web, the lower your zinc isotope values get. Because there's like some isotopic fractionation like in the gut during like zinc uptake in the gut and the intestinal lining, or the absorption of zinc is kind of like different than how it looks like for macronutrient like nitrogen. And so first we have this offset between plants to herbivores. So there we have the isotopic offset, very similar in nitrogen, like the isotope values go up. But once these herbivores are eaten by carnivores, the zinc isotope values go down, down, down. With the exception in hyenas, because you can see this difference between bones and muscle tissue. Most carnivores eat muscle tissue. It's a good idea if you eat someone else to eat the good parts. But hyenas actually eat also a lot of bones, and they look really, really weird in zinc. So let's have a look at those bone-eating carnivores. But zinc, similar to calcium, is a really, really cool new system to look at trophic levels. So meat consumption, differentiating herbivores from carnivores in past ecosystems where we really just have a lot of questions about that. There's also some indication that we might be able to use zinc isotope analysis to look at breastfeeding and weaning, although that is still a little bit in the air. We need way more data for that. So let's have a look at zinc in a second, but here I just wanted to say something. So Clairvia and Vincent Banter's lab, they developed this approach. The lab work for that is quite tedious, extracting zinc from sample materials like tooth enamel. It's very tedious. There are tons of potential contaminations. So Clairvia actually showed that even just wearing normal nitrile gloves is a huge point of concern. You can contaminate your samples just by wearing nitrile gloves, because apparently they contain zinc. I did not know that, or nobody knew that. And it's a little bit frustrating now that since Clairvia did this incredible work for her PhD, has published incredible work, and now we can also measure nitrogen isotope analysis, nitrogen isotopes in tooth enamel. So to some degree, it makes zinc and also like calcium a little bit obsolete. We actually don't need it anymore as a system to tell something about trophic level and meat eating, because we can just do nitrogen isotope analysis. But right now, there are only 2 or 3 labs in the world that do nitrogen isotope analysis in tooth enamel. So whereas zinc, more labs can probably do that. So keep that in mind. Okay, so let's look at the most sexy study that Clairvia has published over the last couple of years. This was published in 2022, where they looked at Neanderthals and associated fauna from a site in Spain. And so, of course, they could have also just done carbon and nitrogen isotope analysis, but I think collagen preservation was probably not very good, so maybe they could not do the classical nitrogen isotope analysis here. But I just want to show you the data. So we have here the Neanderthal, and up here we have some grazers, browsers, kind of mixed feeders here. The bear is always like a mixed feeder. And then here we have like those carnivores, including wolves as an apex predator. And so keep in mind, the higher you are in the food web, the more meat you eat, the lower your zinc isotope values. And so it's really, really cool that Clairvia could show here with her team that, again, like Neanderthals are these apex predators. They apparently eat more meat than a wolf, which I don't understand, right? Makes no sense, right? Like a human, right? Like how, as a human, you need so many nutrients actually from plants. And we know that Neanderthals were eating a lot of plants and they were cooking and all of that. But apparently they are highly, highly carnivorous. Pretty much at any site you look at, that is the case. But here I just wanted to show you, so for generally like zinc isotopes, again, if you have a whole species community you can look at, super valuable to tease apart how much meat actually animals are consuming and depending on. I always find in this graph super interesting how much variation there is actually in wolves. I don't know why I personally find that so interesting, but I find it quite interesting because you would think that they all are adapted to eating a highly meat-biased diet, right? But apparently there's something interesting going on here. And then I also wanted to point out here the crocutas, so like the hyenas. Hyenas pretty much at all of the sites that have been studied so far, modern environments, but also fossil species communities, they always look like that. They always fall with the herbivores. And it is because they're eating a lot of bone material that fractionates differently compared to muscle tissue. And so that is actually very, very interesting. So if you ever have a question about that in your own projects, that you have like maybe some weird animal that has gigantic teeth, and you go like, oh, maybe they're eating bone, zinc can actually tell you the answer to that question, which is kind of unexpected, but that's how it is. Okay, then let me get to the last isotope I wanted to mention, which is calcium. So calcium isotopes are really interesting. Actually, calcium has a ton of different isotopes. The most abundant one is calcium 40, but then we have calcium 42, we have calcium 44. And the most, oh, this graph looks really, really bad. Oh no, it's getting sharper. Okay. So in calcium, because we have so many stable isotopes, some colleagues are measuring calcium 44 and compare that to how much calcium 42 is in a given sample. And some studies are working with the ratio between calcium 42 and calcium 40. And that's why we have to actually like spell it out. Okay, so you will find that may be a little bit confusing, but now you know why. So most colleagues nowadays work with calcium 44 over 42. And calcium is really cool to look at because it also has the same trophic level effect as I just explained in zinc. So again, there's some weird fractionation happening during calcium uptake in the gut. So the higher you are in the food web, the more carnivorous you are, the lower your calcium isotope ratios get. But the cool thing is that calcium, because there's so much calcium in tooth enamel, we can really measure it in small amounts going way back at the time. So a lot of the publications you will actually see from Vincent Baltaire's lab, again, like this is the colleagues from University of Lyon in France that are mainly doing work like that, but also in Germany, Thomas Tütgen is one of the big ones there. It's often applied to cetaceous and Jurassic materials, and dinosaur related research. And this year is kind of like a fun one because here they looked at Tyrannosaurus specimens from a given region in comparison to sympatric herbivorous dinosaurs, and could really show that we have like this cool offset, right? Like so, carnivorous isotopes are carnivorous species have lower calcium isotope values than their herbivorous counterparts. So maybe like comparing that, you know, with Tyrannosaurus is a little bit, you know, stating the obvious. But in some fossil specimens, we really just don't really know, like how they actually relate to other species. So just to mention that here, you can also find a lot of studies where they use calcium isotope analysis to look at breastfeeding and weaning. It's also because they remember milk. You learned in school that milk has a ton of calcium, right? So there's a very, very strong association with calcium also in the archaeological and fossil record. One last calcium paper that I want to show you here. So this is also from Vincent Baltaire's lab in Lyon, where they looked at fossil hominins from East Africa now. So we have, also South Africa. So we have Paranthropus robustus and Paranthropus boisei, so South African and East African hominins. And in comparison with other carnivorous and herbivorous, so this is browsers and grazers, species from a variety of different sites. And here we can see the same thing as we saw earlier with zinc. The higher you are on the food web, the lower your calcium isotope values get. So here the interesting thing is that Paranthropus boisei, I didn't mention that earlier, but Paranthropus boisei has really weird carbon isotope values. So it looks like Paranthropus nutcracker man, you know, these gigantic molars. It was always like the biggest mystery, like what these guys were actually up to, like what were they eating? because it looks like they were eating a lot of C4 plants, and the only C4 plants that we have in these environments in East Africa are like grasses. And then do we have someone in our family tree that is large-brained and is adapted to eating only grasses? Like that actually does not make sense. You know, you don't have enough caloric intake from just eating grasses, even if you eat seeds only. And so that was like always like the biggest mystery, and it remains a gigantic mystery because now we have calcium isotope data from Paranthropus boisei, and it is completely different from any other hominin that has been measured so far. So it's not meat, right? Because like otherwise Paranthropus would be down here. So Paranthropus is not eating, you know, C4 grazers, like zebras and stuff, but Paranthropus is eating some weird plants. The big problem here is that a lot of labs are measuring a lot of, you know, skeletal material, fossils, but very few labs are actually investing into first establishing these plant isotope baselines. So first, like really finding out how does calcium isotopes actually vary in different plant foods. So there might be huge offsets actually between tubers and in comparison to, for example, plant stems, leaves, fruits, seeds. And so I'm actually working with one of Vincent Belter's former PhD students. Her name is Danae. And we will actually be now measuring a ton of zinc, but also calcium isotope values in chimpanzee plant foods. Just like measuring everything, plant foods, but then also insects like termites, ants, and mushrooms, because I'm really into mushrooms these days. Okay, so in summary, we can say that I hope you learned something about like all these different isotope systems. They all have something to bring to the table, yeah? To study past ecologies. And all of these isotope systems that I talked about today can be measured in tooth enamel. So we can really look into deep time because hydroxyapatite, the enamel is made obviously out of hydroxyapatite, and it's like so densely mineralized that we normally are not too concerned about any diagenesis and contamination of these elements like from the burial environment, okay? So if you have a preserved tooth with good-looking tooth enamels, pretty certain that you can measure these isotope systems I discussed today in exactly that material without having an issue with contamination. Generally speaking, these isotope systems we discussed can be a really powerful tool to gain insights into past animal communities, into how the environment looked like and how these animals were associated with each other in terms of food webs. And particularly when we combine these different isotopes. So I showed you some examples where we combine carbon, oxygen, strontium, or carbon and nitrogen and oxygen. So the more we actually combine different isotope systems, we can actually like get a better picture of how the ecology and also the behavior actually looked like in past taxa. And that is pretty much everything that I want you to take away from today's lecture. And I would love for you guys to throw a couple of questions at me if you have some. And if you don't dare, maybe to say it in English, like I'm sure everybody would be happy to translate, okay?